Well, it doesn't surprise me at all that there is this big of a turnout for this particular defense. Um, uh, Micah Marty grew up and went to his undergraduate to get his undergraduate education in the same state and city that I grew up in, Minneapolis, Minnesota, and in fact went to the same high school I went to, which is kind of an odd situation, but there you have it. So, Michael went to St. Olaf to get his undergraduate degree, where he had a whole series of wonderful experiences before he even started graduate school, as very often those kinds of institutions, those really wonderful liberal arts institutions can provide. He went to the Great Barrier Reef. He worked in the Bahamas and in Florida. So he had all of these wonderful experiences even before he started graduate school. And communicated with me about starting in my lab, and I was very happy to have him. And he's been in my lab since May of 2012, so this is his 5th semester, pretty much normative time for finishing a master's. What can you say about Micah? I mean, sunny attitude, quirky sense of humor, unfailingly polite, well-dressed, all the sorts of attributes you want in a graduate student. He's been a pleasure to have in the lab. He really has. A wonderful person through and through, and really distinguished himself in the time that he was here in his desire to teach and to mentor undergraduate students to a degree that I've never seen before. So Michael was really phenomenal in that respect. At the same time that he was doing his graduate work, he was returning to Panama to work over Christmas breaks, doing research on his own and with people that he had established collaborations with in the past. Micah is on to a new adventure immediately after this defense, essentially. Do you leave today?
I leave today.
So needless to say, there doesn't seem to be time for a big party, so this is it, which is nice because you all are here for this particular part of the process. He is going back home and then flying to Naples, Italy, where he is doing a fellowship at the University of Naples with collaborators of mine in the Department of Pharmacy. And he will be working more on the chemistry of sponges, but in this case more oriented towards the applied aspects of chemical compounds and their potential uses as medicines. So he has 6 months and maybe more to work there. Yeah, so that's pretty much all I have to say. Thank you all for coming. It's always wonderful to have a good turnout for a master's defense. And Micah?
Thank you, Joe.
Do I have to turn something off?
The far light switch, that would be nice. Thank you. Thanks for that kind introduction. And thank you all for taking the time to come out to my thesis defense today. I really appreciate your attention and interest in my work. Before I get started, I want to make some acknowledgments. I want to acknowledge Joe. I'm very grateful to Joe for having supported me for the past 2.5 years and mentored me through this process. I'm also very grateful to my committee members, Dr. Jim Blum and Dr. Patrick Irwin, for the support that they've provided to me as I've gone through this process as well as my lab mates, past and present, Professor Sven Zea, a professor from the University of Columbia, who was here for the past year on a Fulbright Fellowship and taught me a lot about sponges. John Vicente, another spongeologist, a grad student at the University of Maryland, as well as the department and the CMS staff and the facilities that I've been privileged to work at for that time. My funding comes through NSF grants to Joe, and I'm grateful to all of these people for their contributions. I'd also like to acknowledge my undergraduate research assistants. These 4 students have made substantial contributions to the collection of the data that I will show you today, and I'm very grateful to them for what they've helped me accomplish. So my master's thesis tests a hypothesis about natural selection by exploring interactions between sponges and their predators on Caribbean coral reefs. Natural selection operates on the traits of organisms, making certain phenotypes more or less common within a population as a function of the survival and reproduction of the individuals that possess those phenotypes. This process is driven by various selective pressures, and among the common biological selective pressures that we think about. We've got predation, disease, competition, parasitism. There are also all sorts of different abiotic pressures that can influence natural selection. But for my talk today, I'll be dealing with the case of predation, so we'll just focus in on that. I want to acknowledge that predation has, there are different ways that predation can play out in affecting a prey population. Predators can either exert direct consumptive effects on their prey species by consuming them directly, or additionally, they can also exert non-consumptive effects. In this case, the lion is modifying the behavior of the wildebeest in ways that may be ecologically and evolutionarily significant. I won't be dealing with any sort of non-consumptive effects, but I want to acknowledge that those are also things that play roles in predation pressure. So let's think about an example of predation as a selective pressure. Here we've got a population of prairie dogs, and for some trait, there are three different phenotypes. We've got the red triangle phenotype, the blue circle, and the yellow square. The red triangle phenotype, as you can see, is kind of a fat prairie dog. This is the fat phenotype, and it is not capable of running very fast. That's what we need to know about this system. We don't need to dwell on other details. So here we've got a population of these prairie dogs, and you can see each of these individuals possesses a different phenotype of these traits. The red triangle phenotype, again, is the fat prairie dog that can't run very fast. You'll notice that the different phenotypes are distributed somewhat evenly among this population, and this trait varies within the population. When we introduce a predator to this system that chases around its prey and catches them by running after them, let's say Fantastic Mr. Fox, and he brings his family along, and so a whole family of foxes are chasing around these prairie dogs, and before long, we have a situation where the fat prairie dogs, the red triangle phenotype, has more or less been removed from the population. It's the fraction of the population that possesses that phenotype is dramatically reduced. So this is a case where predation has exerted a selective pressure on the prey population. The prairie dogs are still doing fine. This population of prairie dogs still exists. They're happy, they're healthy. However, this phenotype has been dramatically reduced. And the fact that there were multiple different phenotypes for this trait allowed this population to adapt to changing environmental conditions, in this case, the arrival of a new predator. But what about organisms that can't run in any phenotype, like a tree or sessile benthic invertebrates? Well, these organisms have various strategies for surviving predation. One possibility is behavioral escape. In this case, we've got tube worms that hide from their predators when they feel threatened. You can see they retract down into a tube to get out of the way of the predators. But if you can't run and you can't hide, sometimes you have to stand your ground, and some organisms have defense mechanisms that they use to stand their ground despite the threat of predators. One type of defense that organisms use is physical defenses. Here we've got some plants that have spines on them that prevent herbivores from getting to the foliage. In this case, we've got a clam that has a shell that protects it physically from predators getting at the soft tissue inside. Another strategy to dealing with this is chemical defenses. So chemical defenses are widespread across taxa. Here we've got examples from sponges, ascidians, octocorals, algae, and terrestrial plants. And with anti-predatory chemical defenses, you have these structurally complex secondary metabolites that the organisms have in their tissues. In some cases, they may be made by symbiotic microorganisms. In other cases, they're made by the organism itself. But these chemicals either reduce the fitness of the organism that feeds on them, or they actually deter the organism from feeding on them entirely by tasting really bad. We'll focus more in my talk today on the tasting bad side of those. But I want to emphasize that this is sort of common across different taxonomic groups. One group where this is particularly well studied and the ecology of these chemical defenses is well established is with Caribbean sponges. It's probably no surprise to this audience that Dr. Pollack has done a lot of work on this subject, and we've got a very clear framework for understanding the chemical ecology of Caribbean sponges. We organize sponges into three discrete categories according to their chemical defense. We have preferred sponges, which are groomed entirely off of the reef and found only in refuge habitat like mangroves or under rocks on coral reefs where the predators can't access them. We have palatable sponges, which don't have this deterrent chemistry in them. However, they persist on top of the reef because they've got exceptionally fast growth rates and things like that. And then finally, we have defended sponges, which persist on the reef because they're deterrent and the predators don't want to eat them. We know the secondary metabolites that are deterrent to fishes for a number of defended sponges. And we've evaluated, Joe did work back in the 90s to evaluate the chemical defense strategies of a whole slew of sponges from the Caribbean. You'll notice if you look at these data that there are two discrete patterns. I won't describe how these data are generated just yet. Later in my talk I'll address this because this is the same method I use. It's a bluehead feeding assay. But the way to read this chart is that large bars like this indicate palatable sponges. Small or absent bars like this indicate defended sponges. Across all taxa of sponges in the Caribbean, we see this general pattern of some sponges being defended and other sponges being palatable to their predators. And this system, the predators that are feeding on them, the most ecologically important predators are spongivorous fishes. We've got angelfishes in the top row, parrotfishes here, and then trunk, file, and boxfishes at the bottom. And these fishes rove around on the reefs, taking small bites out of the sponges as they go. The effect that these fishes have on sponges is very well established through rigorous empirical tests. This study here was a growth experiment that was conducted in the Florida Keys. In the top panel, we've got Calispongia armigera, a palatable sponge, and in the bottom panel, we have Amphimedon compressa, a defended sponge. And you'll notice that there are two different treatment groups. There's inside cages and outside cages. And the sponges that were grown inside of cages for Calispongia armigera grew much more than those raised outside of cages. And this is because outside of the cages, the predators have access to them and can feed on them and reduce their capacity to grow. With Amphimedon compressa, we actually don't see this difference because Amphimedon compressa is chemically defended. And what this means is that the predators are both not feeding on Amphimedon compressa and its growth rate is significantly reduced. You'll notice that on the axis here, Amphimedon compressa grows a lot less both inside and outside of cages than Calispongia armigera does. This was a reductionist experiment that basically told us there appears to be a cost to chemical defenses, and those that are not defended are clearly reduced in their ability to grow by spongivorous fishes feeding on them. Now, these results were magnified by doing a large biogeographic comparison. On the axis here, it shows a bunch of different sites around the Caribbean region, and this y-axis is indicating these white dots that descend as we go along. And that's the abundance of spongivorous fishes at these sites throughout the Caribbean. As the abundance of these predators declines, the proportion of the population made up by palatable individuals increases. So what we see is when the predators are absent, the community shifts to have more of these sponges than these sponges because they grow fast and are relieved from the pressure of predation. We see a large-scale community effect of the predator removal. Now, within this system, I was saying that we have defended sponges and we have palatable sponges, and that segregates out by species. But there are some species that are variable in their chemical defense. Here are a couple examples of this. This is a situation where some individuals within the species are defended and other individuals are palatable. So we can think of this like two different phenotypes. When we populate a population with these organisms, we can see there's a whole bunch of, we have a population of variably defended species. This is sort of what it looks like. This is all one species. The dark individuals are defended, the light individuals are palatable, and overall, this population is variably defended. So the overfishing of sponge predators throughout the Caribbean provides an opportunity to test a hypothesis about natural selection and how it acts on this variable chemical defense trait. So we assume that there's a metabolic cost to chemical defense. We saw that in the growth experiments across species, and within species, we have reason to suspect that these palatable individuals here that are not synthesizing chemical defenses, not storing chemical defenses, are going to have the capacity to grow faster than the defended individuals. And then we also assume that there's a non-random selective pressure exerted by the predators on the prey population. Assuming these things, we would expect that the removal of the predators would cause the population to shift to have a larger fraction of individuals who are palatable to the predators. So my hypothesis is that populations of variably defended sponge species will be more palatable and exhibit greater variability in the chemical defenses on overfished reefs where spongivorous fishes are absent. To reiterate this with another cartoon, again here, the palatable individuals are white, the defended individuals are dark, and the top row here is an overfished reef through time. The bottom row is a protected reef through time. You can see on the protected reef, the spongivorous fishes remain, and the population stays about the same. However, on the overfished reef, as the predators disappear, we slowly see a greater fraction of the population being composed of the palatable individuals. This is what I'm hypothesizing to see, and set out to test this hypothesis. So how did I do that? I chose 8 variably defended sponge species to look at from the Caribbean. I actually have to exclude Spatiospongia vesparium due to some funny taxonomy issues. I'm just not including that in my data and my analysis for my thesis. But these 7 species are the sponge species that I worked with. You can see that they vary considerably in their morphological habits. They represent a variety of different ecological types of sponges. And I collected these from a variety of sites around the Caribbean because I wanted to get a sense of what the populations look like under different predation regimes. So sponge collection occurred at a site. I would try to collect about 10 individuals from the population of a certain species of sponge. I tried to get as many species at as many sites as possible, but I don't have a completely homogeneous distribution of these things. With each sponge that I sampled, I took 10 milliliters of sponge tissue, and I placed it as soon as possible into extraction medium of 1:1 dichloromethane and methanol. At the same sites, or at very close nearby sites, Lowe and Pollock conducted fish surveys. These are just standard belt transect surveys on scuba that measured a 2,000 cubic meter volume of water. And along these transects, they identified and enumerated the spongivorous fishes that were there to generate a spongivore index. And the spongivore index basically just says the most important spongivores added together, divided by a correction factor that takes into account local differences in the size of the sponge predators. So I use this as my metric for determining what the selective pressure looks like at each of these sites. And I collect my own sponges to get my own sense of what the chemical defense looks like at each site. Back in the laboratory, I followed a procedure the Pollock lab has been using for the past 2 decades to assess the anti-predatory chemical defenses of marine invertebrates. It basically involves a chemical extraction procedure and then a preparation of the chemical extract into food pellets that are then fed in feeding bioassays to yellow-faced bluehead wrasse. These images here depict the process. I had the privilege while working here to put together a video manuscript with Joe showing how this assay is conducted and how we do the extractions. The first 6 panels indicate the extraction process. I take sponge tissue, I put it into the solvent, I agitate it for a while, and then some glassware and rotary evaporation takes place. And at the end, I have a crude dried organic extract of sponge tissue. It's basically just the dried flavor of the sponge. And I know the volume of that initial sponge. So then I can reconstitute that sponge into a volume, an identical volume of a nutritionally comparable food matrix that's made of squid mantle powder and sodium alginate as a gelling agent. And then I take this, I mix it up, I make a homogeneous mixture of sponge extract in this food matrix. I make little noodles with it, and I chop the noodles into little pellets that I can then feed to the fish in the assay. I make control pellets in exactly the same way, except I do not add any extract to the control pellets. So the assays take place down at CMS. I have multiple laboratory aquaria that I keep these yellow-faced bluehead wrasse in, and I keep 3 fish per cell, and each cell has an opaque divider so that the fish can't observe what's going on in the adjacent cell. That makes these independent replicates. The fish can't see that I'm offering a sponge that tastes really bad to the previous group and say, well, I'm not going to sample that. These are independent replicates, and I do 10 independent replicates for each sample. So every sponge I collect generates 1 extract, and that extract gets fed to 10 different groups of fish to evaluate whether they accept it or reject it. This shows a video of accepting a pellet and rejecting a pellet to give you a sense of what that looks like. This is when the fish accept a pellet. You can see they grab it, they take it into their mouth cavity, and it doesn't come back out. The response is very clear with this rejection here. You can see that they repeatedly sample the sponge, the pellet, and repeatedly reject it. So the determination of this assay lies in seeing whether they accept a pellet or whether they reject a pellet. This flowchart demonstrates a single run of the assay. First, a control pellet is offered. If the control pellet is rejected, we can't know why they rejected the control. We can't work with them. They're not going to be a cooperative group of fish. If they accept the control pellet, we offer a treated pellet. If the treated pellet is accepted, then the sample can be scored as accepted. There was nothing in the extract of this sponge that made it unpalatable to these fish. However, if the treated pellet is rejected, then a subsequent control must be offered because they may have rejected the treated pellet simply because they were full. They didn't want to eat anymore. So if they reject the subsequent control, they're uncooperative. We can't use those data, and we work with a different group of fish. But if they reject a treated pellet and then they accept a subsequent control, we can say that we know that the only difference between this control and that treated pellet is the fact that the treated pellet had the sponge flavor in it. So the 10 replicate runs of each sample generate a number of pellets eaten out of 10, and that's a metric I can use. I plot it on the y-axis over here because most of the graphs you'll see in my results here have that axis. And these assay data quantify the chemical defense levels for an individual sponge. I tested multiple sponges from each population, so I can generate a mean number of pellets eaten for each population, and I can calculate the standard deviation around that mean number of pellets eaten, and I can use that standard deviation as a measure of the variability of this chemical defense within that population. So for my analysis, I compared these data to the spongivore index data, the spongivore abundance data from Lowe and Pollock in both linear regressions and logistic regressions. I use these two different types of analysis to get at two slightly different questions. The first one, I'm using a linear regression to assess the standard deviation. This is my effort to look at the variability of the chemical defenses. I look at the standard deviation of a given population and where that population falls out on the spongivore index. And hypothetically, I'm expecting that there's a higher standard deviation at low values of the spongivore index and lower standard deviation at high values of the spongivore index, because over here, there's a selective pressure of predation that's keeping all of the sponges within a certain chemical defense range. Whereas on overfished sites, that pressure has been relieved and the palatable individuals can persist in the population. The other component of the hypothesis that I'm testing is using a logistic regression, and I'm comparing the number of pellets eaten in the assay to the spongivore index. So this is looking more at the overall palatability. And in this case, I'm not working with just populations. I'm working with individual observations. So the logistic regression model can deal, it works with binary data. So in an individual run of the assay, a pellet is either accepted or it is rejected. And this model deals with binary data of that sort, and it uses this sigmoidal curve, which is perhaps more appropriate for what our expectations are for the way these data behave. And so, like I said, the linear regression is sort of getting at the question of how variable is each population, and the logistic regression is getting at the question of how palatable is this population in general. They're both using spongivore index as a predictor. This is overall what I'm sort of trying to assess, whether or not the predators are having an influence on these chemical defense traits within the prey. So here are just the raw data. This chart's overwhelming. There's a lot to look at here. I just want to show it to you so you have an understanding of what the raw data that I work with in the rest of this talk looks like. On the y-axis here, we have the number of pellets eaten in the assay. That's a number out of 10. And down here, we've got the spongivore index. The first panel is all species combined together, and the remaining panels are individual species parsed out on their own. You can see that for some species, I don't have tremendous site replication, but just sort of qualitatively across all of these, you can see that the data are very well spread out. At very low values of the spongivore index, we sort of have the full range of potential outcomes for the number of pellets eaten. And at the high end of the spongivore index, we also have the full range of potential outcomes, which gives you a sort of first glimpse into what my results look like. Now, this figure is very overwhelming. I want to walk you through it really slowly, so bear with me. On the far left axis, we look at the number of pellets eaten in the assay. This axis is representing each of these box plots here. So this box plot, for example, is ranging from 2 to 5 approximately, and that's in number of pellets eaten. For the box plots, the means are drawn as open white circles inside of the box plots, and the box plots are organized according to ascending mean values. You'll see at the 6 on this axis, there's a dotted line that runs across. That's the significance cutoff for these type of palatability data. If a pellet is, if an extract is rejected, is accepted 6 or fewer times when I run the assay, that extract is considered deterrent. If it's accepted more than 6 times, it's considered palatable. So this is the cutoff line that we traditionally have for looking at whether an individual is palatable or defended. On the right side here, I have the sponge-bore index plotted, and this is represented by these triangles, which are artificially connected to one another. This is just to give you a qualitative sense of how the sponge-bore index looks in relation to the trends of palatability within among the populations. And then on the horizontal axis here, I have all of the different sites at which I collected these. The sample size of the site is represented in parentheses, and the first one here is actually our previous observations. So these are from Dr. Pollack's work back in 1995, or potentially the update that came out this past spring by Lowe and Pollack. At the very top, we have bars that indicate the results of analysis of variance and a Tukey post hoc test. The bars indicate populations that were not significantly different from one another. So, for example, this bar indicates that this population was not significantly different from any of these populations up until here. So we see a gradual trend of significant differences among the populations, but one thing I want to point out is that all of these box plots fall approximately within the range of the previous observations. It's quite remarkable that the previous 4 observations caught the whole variability that I caught in about 120 extra assays. One thing to note is that these 4 populations up here, they're composed exclusively of palatable individuals, but not in accordance with the hypothesis. 2 of them are at very high sponge-bore abundance sites, but I'll get into the results of my actual analysis in a moment. This is just sort of a qualitative look at these patterns. The next species here is Iotrichota birodulata. The graph is organized in the same way. One thing I want to point out here is that these 3 populations are exclusively composed of defended individuals, which is very surprising because our previous assessments showed a much more palatable version of this sponge. And so these results differ from previous results in that way. We also have some slight differences as we go along here, but again, qualitatively, they don't seem to match very well with the observations of the sponge-bore abundance. Now I'm getting into the individuals where I have the species where I have a few less replication. This is Clathria virgultosa. There's very low replication at each of these sites, so we don't really have much by way of box plots, but this does fall approximately within the range of previous observations. Cribriplina basculum, again, falls sort of within that range. It is definitely much more defended than previous observations, as you can see by the mean values. The mean here is above the cutoff. The mean here is below. However, all 3 of these populations that I surveyed did have some individuals that were palatable. So they still sort of fall within this, they have variability in this trait. The last 3 species that I'm going to show you this type of box plot for are plotted here: Desmopsima anchorata, Dragmastodon reticulata, and Zestospongia muta. With Desmopsima anchorata, we actually see the pattern that the hypothesis predicts. At a site with lower spongivore abundance, we see a greater palatability than a site with higher spongivore abundance. So that does conform to the hypothetical expectations. Dragmastodon reticulata, we really don't have good insight into this because we just have 1 population. However, we do see quite a bit of spread in the data at a site with high spongivore abundance, which is not consistent with my hypothesis that at high spongivore abundance we will see less variability within the data. Zestospongia muta here is the population in Bocas is more palatable than some of our previous observations, but again, this is a hypervariable sponge, and we can't say much with just 1 observation like that. So moving on to linear regression analyses, Again, that's a very busy slide. Let me walk you through it. This is the linear regressions. We're looking at the relationship between variability and predators. The variability was measured by the standard deviation around the mean number of pellets eaten and is plotted on the y-axis here. The spongivore index is the metric of selective pressure. It's the metric of predator pressure, and that's on the x-axis here. And this first one is all species combined. Now, what I want to emphasize about these points is that each point represents a distinct population, not a single individual. This is the standard deviation around the mean number of pellets eaten for a single population and the abundance of the predators at that site. So for all species, this includes all of the species shown in pictures here, and the replication for each species is depicted in the bottom corner of the picture. So Iotrachota had 7 populations, Niphedes had 14, etc. You'll notice by the p-value and the r-squared here that we did not find what we were looking for. The p-value is not significant and the r-squared is really poor when we look at all species. The same is true when we look just at Niphedes digitalis. Again, we have this negative trend, but it's not significant and the r-squared value is 0.04. There's not a relationship that we can really see here. Same thing is true for Iotrachota biarticulata, an insignificant p-value and a poor r-squared value. And again, for Zestospongia muta, we have this same pattern. The trend is stronger here, but just to contextualize this point right here is the second highest in the response variable, and yet it's the highest in the predictor variable, so that makes no sense. It does not conform to what we're expecting. So overall, the linear regression analyses did not show a significant relationship between the variability of chemical defenses and the predator abundance. Onto the logistic regression models. Here we actually did see some significant patterns. So I'm going to unpack this for you slowly. The logistic regression model used spongivore index as a predictor variable. It modeled the probability of assay fish accepting a pellet. And then each of these separate designations is a separate run of the model. So first the model was run with all species, and then with each of these species individually. They're shown in pictures here alphabetically, and they're organized alphabetically there so you can see how they're related to one another. The number of populations that went into the model for each species is shown in the bottom corner of the picture. All species was highly significant, and then within each of these species we had variable, sort of inconsistent patterns. Clathria virgultosa, Desmopsima anchorata, and Niphedes digitalis were all significant, as indicated here by the darker box around the photo. However, the estimate, the directionality of this influence for these two sponges, Clathria virgultosa and Curvicula vasculum, is in the wrong direction of what we expect. In this case, as the predators increase, so does the probability of the fish accepting a pellet. They become more palatable as the predators increase, which is not, doesn't seem like a realistic case. In these three cases, they all become less palatable. However, Iotrachota biarticulata is not significant. Overall, we have sort of inconsistent results here. But the point that I really want to emphasize is that these estimates that we're looking at are incredibly weak. This does not mean the predictive power of this estimate, not the predictive power, but this estimate tells us that the effect that the spongivores are having on the palatability, if they are having an effect, is incredibly small. So to contextualize this in sort of ecological contexts that make more sense in our system, we ran a multi-category logistic regression model. Now, this differs from a standard logistic regression model in that you can specify multiple categories. With a standard logistic regression, it's a binomial response, so there are 2 categories. Here, I specified 3 categories. I assigned sponges into categories of no defense, intermediate defense, and high defense, and modeled the probability of the spongivore index causing a shift in that defense category from one category to another. And the directionality that I specified was that the spongivore index would cause them to become more defended. So the results of this model are not significant. Basically, the spongivore index cannot exert a significant effect on the palatability of these sponges to cause them to shift into a new defense category. To give you a sense of what this actually looks like, the estimate here for all species, this value, when you plug it into the predictive equation of the model, it says that it would require a change in 150 units of the spongivore index. That's approximately an addition of 150 spongivore species to a reef site in order to affect the smallest magnitude of change into a new defense category. Now, in practice, in the Caribbean, the mean value of spongivore index is 28. And the maximum value that Lowe and Pollock found in the Caribbean was 85. And so a change in units of 150 is just not practical. It's not realistic. That could never happen. So in summary, the linear regression analyses exhibited, in all cases, a weak relationship, a negative trend, and an insignificant p-value. For the logistic regression model, we had inconsistent results. The trends were inconsistent, the relationship was always weak, and some of them were significant, others were not. For the multi-category logistic regression model, we had a weak relationship in all cases, the trends were inconsistent, and the p-value was not significant in all cases. So overall, what does this mean? We found poor, weak, and inconsistent support for the hypothesis. Predators do not, cannot be, the flavor, the palatability of the sponges cannot be accounted for by the predator abundance. Why might this be? There's a possibility that associational refugia are muddling this picture for us. So I'm not going to steal Michael Wooster's thunder. He's going to give an honors thesis talk next spring on this subject, but he's been looking at associational refugia in sponges. This basically is where a palatable benthic organism will live in close association to some sort of defended or structurally complex other benthic organism. So in this case, we've got an orange palatable sponge living inside of this coral head, and it lives there just fine. It survives, but it's not accessible to its predators. In this case, we have another palatable orange sponge living among some other sponges and some reef rubble. But basically, there's the possibility that these data are muddled a bit because the palatable individuals of the population at a site with abundant spongivores are living in associational refugia. Here, this chart shows the 7 species that I worked with and all of the data collected on them combined into single box plots. And they're plotted alongside the 2 most common defended sponges from the Caribbean and the 2 most common palatable sponges from the Caribbean. There is clearly a distinct group here of these variably defended individuals, these very variably defended sponge species. They are clearly distinct from the defended and the palatable species that we've got, which begs the question, could this be some sort of alternative adaptive strategy? One possible explanation that could give rise to this being an adaptive strategy is automimicry. So in automimicry, this is basically just where the chemical defense has multiple phenotypes. There's some individuals that are defended and others that are not, referred to as automimics. When the predators come along, they can't tell the difference among the individuals. So all that from the experience of a predator, this just incurs a cost of extra handling time because foraging involves sometimes sampling something that tastes bad and having to reject it, and sometimes not. So if there are other benthic organisms that are nutritionally comparable and don't have this chemical defense, it's possible that simply having variable chemical defenses allow these sponges to avoid being predated upon. It's a possibility. In conclusion, I found weak and inconsistent support for my hypothesis. Along the way, I gained lots of factual information about these variably defended sponge species, so we now have a stronger understanding of what their approach to chemical defense looks like. It may have opened some avenue for exploring the potential for variable defenses being an adaptive strategy, but I really want to emphasize that this is a case where obvious theoretical predictions fail. Natural selection does not necessarily optimize organisms for all aspects of their environment. And as biologists, it's an important lesson for us to recognize that we are not always going to find pattern when we look for it in nature. With that, I thank you for your attention, and I invite any questions.